Good morning, everyone. This is Shane. I'm a postdoc research fellow from Dr. Joseph Wu's lab at Stanford Cardiovascular Institute. Today is my pleasure to provide the updates from cardiotoxicity working group. Yeah, firstly, our mission is to select the in silico and in vitro tools that can accommodate the complex mixtures represented by the botanicals. Secondly, we would like to select the candidate botanicals based on the suspected toxicity or safety with respect to the cardiotoxicity endpoints. And thirdly, we would like to establish a series of cardiotoxicity botanical case studies such that we can evaluate the usefulness of growing tubers. So here comes the list of our current working group members. We have the principal investigators and scientists from L'Oreal and Fujifilm CDI, Innovitro, and the FDA and University of Michigan and Stanford University. So here's the list of the botanicals that were selected and then investigated from by the cardiotoxicity group. So on top of the botanicals selected by other working group, we further added iconite, ephedrine, and olinder, tripterogium, and euhenbie, which we suspect that can induce the cardiotoxicity based on the previous publications. Well, the Asian ginseng and milk thistle were highlighted in green that we expect that they do not have any effect. Next, I would like to go over about the assays that we have utilized in our study. So here, when we are talking about the cardiomyocytes, we can appreciate that they can generate a spontaneous beating and they are excitable cells. So if we look at the curve in black here, that which indicate the cell membrane potential changes over the time. So due to the opening of voltage-gated sodium channels and voltage-gated calcium channels, the membrane potential will go from negative to positive, depolarized. And then later, due to the opening of potassium channels, it will bring the membrane potential back to the negative resting membrane potential. At the same time, so if we look at here, that the opening of the L-type calcium channels will allow the calcium ions to move from outside into the cytosol, which will at the same time induce a larger amount of calcium release from the SR through the ryanodine receptor, which we call the calcium-induced calcium release. And then later, after calcium ions reach the peak in the cytosol, and the calcium can be pumped back to the SR by Surcar and move out of the cell by the NCX, the sodium and calcium exchanger, and also the calcium pump. So if we monitor the intracellular calcium concentration changes, we will be able to get a curve like this, showing the blue color, like this. In addition, the cardiomyocytes, they are quite specialized cells. They have the sarcomere structure, which consists of the thin and thick filaments. The calcium ions are able to bind to the cardiac troponin complex on the thin filaments, and then this will lead to the conformational changes. So that's why we will observe the contraction phenomena of the cardiomyocytes. And then as the calcium release from the troponin complex, the cell will become relaxed. So we will also be able to get the contraction and the relaxation curve here. So in summary, that the cardiomyocytes action potential change, and then the calcium transient change, and then the contraction relaxation change are the three key characteristics that we want to when we want to assess the cardiomyocytes function. So here we use the human iPS-derived cardiomyocytes as the model to study the potential cardiotoxicity of these selected botanicals. Human iPS derived cardiomyocytes has been proved to be a very reliable and then just efficient platform to assess the drug toxicity. And then in terms of the assays, in addition, we also add the seahorse assay to look at the mitochondrial function, as well as we also use the HEC293 cell line to look at each individual ion channel and the receptors that are associated with the cardiac functions to see whether these botanicals can affect these channels and then receptors. Next, I would like to go over about how we did measurement in each individual assay. So about the measurement of the action potential and the calcium transient, we firstly plate the cells on the cell culture plates, and then we incubate the cell with the voltage sensitive or calcium sensitive dye, so that the voltage change or calcium changes can be translated into the fluorescence change. And then we did the image acquisition, and then later we can obtain the curves like this. Here we can see that the intracellular calcium change and then action potential change over the time, where we can analyze the data by looking at several parameters. For example, the amplitude, the upstroke decay, and then as well as the action potential upstroke and then decay. For the cardiomyocyte contractile force measurement, so the cells are plated on the cell culture plate, but on the bottom of the plate, there's a PDMS membrane that can bend over. So as the cardiomyocytes beat, so the movement of the PDMS membrane will be recorded, which can indicate the contractile force. So on the panel C, you can see that we can finally get a curve like this, where we can analyze the contraction force, beating rate, or there's any early contraction happen. In terms of the seahorse assay, so we firstly plate the cells on the seahorse assay culture plates, and then treat with the botanical or vehicle control. And then here, after the basal recording of the oxygen consumption rate, and then we challenge the cell with the oligomycin, which is the inhibitor of the ATP synthesis. So here, the difference in the OCR brought by the oligomycin will indicate the ATP-linked respiration. And then next, we'll inject FCCP, which is an uncoupling agent that can disrupt the mitochondrial membrane potential. So the electron flow through the ETC will be uninhibited. So the oxygen consumption rate will reach the maximum. And then later, the retinol and then the antimycin A will be added. So the combination of which will shut down the mitochondrial respiration, so that we can also calculate the non-mitochondrial oxygen consumption. Next, I would like to show some of the case studies. So firstly, the milk thistle. So even though here we expect that it can be safe, but due to look at our data that, for example, the contractile force, the acute treatment, we can see that when it comes to the higher concentration, it can decrease the beating rate when we measure the contractility. And also when we measure the calcium transient, that we can see that some of the concentrations will bring the changes in the upstroke slope, as well as the amplitude. And then we also observe a dose-dependent inhibition of the mitochondrial function by looking at the basal respiration and then spare capacity, maximum respiration, and then ATP production. And Aconite, there's several publications showing that in the animal models, aconite is able to induce the ventricular arrhythmia. So as expected, we can see from the contractile force measurement, the higher concentration will lead to the dramatic changes in the beating duration and then the downstroke slope. And in the calcium measurement, we can see that there's a significant increase in the beating frequency, and also across several concentration tested. And then for the seahorse assay, we can also see that there's an inhibition in the respiration by the aconite. Olinder, according to the previous study, Olinder is able to inhibit the sodium and potassium ATPase activity, so that it will be bringing the arrhythmia into the cardiomyocytes in the animal studies. So looking at our data that when we measure the contractility, we can see that Olinder has a dramatic effect on the several parameters, including the amplitude and then the upstroke slope, downstroke slope, beating rate, beating duration. We also observe effect when we measure the calcium transit. For example, we can see the amplitude has increased a lot and by the Olinder. Also, we see that there's a decrease in the several parameters in the seahorse assay by Olinder, including the spare capacity and then the ATP production. So due to the time, I won't show all the data but we have completed all the assays. So we are on the stage of data analysis and then drafting the data paper. In terms of the 2025 highlights, we published the strategy paper, which was just online last week. And then also we completed all the planned experimental work. So we tested 16 botanicals across the 6 assays. So in consideration of the multiple doses and the multiple time points, this is really an incredible amount of work. So thanks all for all the team members. And also, as I mentioned, that we have began to outline and then drafted the data paper. And then we've discussed the other analysis, like using the benchmark concentration modeling to understand the human responses. Here, I would like to show that the paper, strategy paper from our cardiotoxicity group, which was just online last week, where we describe in detail that how we developed the approach for evaluating the cardiac potential, cardiotoxicity potential of the botanicals. So here, if you are interested, please scan the QR code here on the right. In terms of the 2025 outreach, Dr. Matthias Gelsman has done perfect oral and poster presentations in multiple conferences. For example, the SOT and the Eurotox and MPS and SPS. So special and huge thanks to Matthias for leading the outreach for the cardiotoxicity group for the past year. For the next step, firstly, we would like to make the data publicly available. And then also we can draft and then publish the data paper. Hopefully we can get it ready and then submit in early next year. And then thirdly, we would like to discuss the comparisons and collaboration with other groups. For example, the neuron toxicity and ANMI, because like cardiomyocytes and neuronal cells, they are both the excitable cells. Maybe we can just compare the data and then to see whether they have some similarities in terms of the response of these botanicals. And also, definitely we would like to collaborate and then discuss with the ANMI group because this is important to see the in vitro data, whether it can translate into the in vivo data. Next, we would like also to present and then our data in the 2026 meetings, for example, the SOT 2026. And lastly, we would like to discuss and plan the future work, which could include different cell types, because you know that in the heart, the cardiomyocytes, this is not the only cell type that resides in the heart. That we also have the endothelial cells, like cardiac fibroblasts, and then the smooth muscle cells. So the heart is a complex organ that have multiple cell types. So definitely, if our budget allows, if we can look at the effect of these botanicals on other cell types, that will also be helpful to see this effect on the real human heart. Also, secondly, about the lot-to-lot comparison about botanicals to understand the assay response. Since you know that the human iPS derived cardiomyocytes, maybe we need to include more data by different lot, because lot-to-lot variation is really a problem of this model. And then lastly, if the budget allowed, we would also like to look at more botanicals in these assays. to make the whole study more complete. Okay, thank you so much. This is all the update from cardiotoxicity group.
Great, thank you so much, Shane. We have time for 1 or 2 questions if anyone wants to put one in the Q&A or raise your hand.
While we're waiting, I can just ask Shane, that was really nice. We've generated a lot of data in this team and you weren't able to show all of it, but in your work, you did some that were at different time points. Could you maybe speak, I know you don't have them on slides, but talk about how you did some very early, like 1 hour time points and then later time points, and how, if any of that changed the response of the botanicals? Or is that something we need to dig into? Maybe you don't have it offhand.
Yeah, from our hand that I performed the seahorse assay and then I included 2 time points, the 1 hour and then I included 3 time points, the 1 hour and then 2 days and then 7 days. So I can see that this like time dependent responses. For the other assays for like contractility measurement, calcium measurement, we also include acute treatment 20 minutes, and then there's a longer treatment like 2 days. So I think for we expect the data to be time dependent, and then indeed really observe this. Just how we just include the data into the data paper, we need to further discuss because just maybe 7 days treatment, that is too long for the botanical treatment, and then like 20 minutes is fine because that if there's some inhibition of the botanicals on the ion channels, the ion channel should respond really quickly, usually within several minutes. So 20 minutes, that is enough to see the changes. Also for the seahorse assay, we consider as the like kind of the metabolism, and then just 1 hour is also enough for like acute treatment. But for the seahorse assay, that 7 days, I think is not that too long. Yeah.
Thanks, Shane.
Thanks, Shane. One question from the Q&A was, are there any plans to use cardiac organoids or other 3D models for detection of cardiotoxicity?
Yeah, definitely. As I mentioned in the future plan, that we plan to include the other cell types like cardiac fibroblasts, the endothelial cells. But just if we can utilize the 3D model, that is perfect because the 3D model that mimic the real in vivo heart organization. And then in our lab, Dr. Wu's lab, we recently published a paper in Science where we show that we have a very efficient protocol that we can get a 3D vascularized heart organoid within several days, and then this will be very efficient model, and to see like we do not need to just test the individual cells and then just discuss and to see what is and then just speculate what is the whole response. But just the 3D model give our very informative information. So for sure that is in our plan if the budget allow.